go in completely the opposite direction of Xiaoping's talk. He was going sort of broad scale, top down machine learning, and I'm going to go very detailed about processes. But so I've been recently looking at basically how we can I guess take a look at how we're modeling biogeochemical cycling and think more about elements besides carbon and how they interact with the carbon cycle. And so I'm thinking especially about redox sensitive elements. And so this I'm going to talk about a few different projects which were supported by a mix of the NG Arctic program and an early career award from DOE Office of Science and also some ORNL internal funding. So when we model soil carbon cycling in the models that we use typically at large scales, we're usually starting from a fairly simple starting point, which is first order decomposition models. So these models are basically set up like the equation in this slide, where we have the change in the carbon stock of a pool over time is related to some fixed turnover rate parameter of that pool times functions of environmental variables like temperature and moisture times the size of that pool. So this pool turnover rate is an important factor in this type of model. And it's very sort of focused on carbon and focused on the inherent properties of the organic matter pool that we're looking at. So what's missing from these carbon focused models, and I think I shouldn't say only carbon focused because a lot of the current models are using carbon and also macronutrients such as nitrogen and phosphorus, which are all incorporated into a broader biogeochemical cycle. But what these models are not including is some of these other elements, such as in this example, iron. So I'll first talk about an example from some of the NG Arctic work that I've been doing, looking at how we can incorporate the role of iron cycling in these iron-rich Arctic soils into the simulations that we do of carbon cycling and greenhouse gas emissions from iron-rich soils. So these are some really nice visuals from Beth Herndon. So on the left, there's a nice diagram showing how the iron cycle interacts with the carbon cycle. You have these mineral transformations, iron 3 oxyhydroxides under more oxic conditions, and then under anoxic conditions, they can be reduced from mostly iron 3 to iron 2, and then these interact with the carbon cycle. And this is sort of from the chemistry point of view, it's a lot of interesting complexity, but also just from looking at these soils, you can see the influence of the iron cycle. You have these iron oxides forming in visible amounts on the surface of these flooded soils as a result of this really active iron cycle. So taking a step back, I think something that we're really interested in modeling accurately, especially in high latitude soils, is what happens to carbon in those soils under different situations of hydrology. So for example, under high water table versus low water table conditions, are we going to have emissions of carbon dioxide? Are we going to have emissions of methane? And what's the mix between those fluxes? So here's one example of an experiment which shows sort of our starting point for understanding these processes. The expectation generally is that when a soil is inundated, you have less oxygen penetration into that soil. And because aerobic decomposition requires oxygen under high water table conditions in these white symbols, you have a decrease in CO2 fluxes. And you also eventually have an increase in methane fluxes, which are produced under these by anaerobic processes under anoxic conditions. So there's tons of experimental evidence for this trade-off between CO2 and methane fluxes under high water table versus low water table conditions. And these assumptions are integrated into the models that we use at large scales. This table is from a really nice paper comparing methane models from a bunch of different large scale models. and there's some differences, but there's also a lot of commonalities. These models tend to assume that there's a certain ratio of methane to carbon dioxide, that methane flux is related to heterotrophic respiration, and a lot of them also include this function of water table depth at a particular point in time, which controls essentially whether that soil is inundated, which then determines whether that soil is producing methane more or CO2. So this is a depiction of like the simplest version of that model, where the model says, is this soil inundated? When it's underwater, we're going to produce methane. When it's above water, we're going to produce more CO2. And this is a simplification. Those models are not all that simple, but this is what I'm using as kind of the starting point. So if we look back at our diagram of methane production under high versus low water table conditions, we can see that under high water table conditions, you do have higher methane production, but there's a delay of, in this system, 50 to 70 days before methane production really ramped up. And so one way we can incorporate that is something that was included in the CLM4Me model, which is this FPE, I believe, which is essentially a time delay function, which represents the depletion of alternative terminal electron acceptors. So that's an empirical approach to representing this type of delay. But I think also, particularly in these soils where we know that the iron cycle is important, we can try to take a process-based approach to sort of see if we can integrate a different cycle with the carbon cycle to explain this pattern. So we know based on observations that in these high iron soils, there's a relationship between methane and the oxidation state of iron. So higher iron 3 concentrations are associated with lower methane concentrations. That suggests that there's an interaction there. There's also some evidence from incubation studies where if we look over time, there's this increase in the concentration of iron 2, which is produced by iron reduction processes that microbes can use to support decomposition. And this is more or less correlated with a change in the ratio of CO2 to methane production over time, which the amount of CO2 per unit methane declines over time as the iron concentration is changing. So in terms of modeling this, I was able to put together a reaction network, including carbon and iron reactions. This is set up in the p-Flowtran model. And so the way that this model is depicting these processes is we have some soil organic matter pools that can be depolymerized into soluble DOM. And then DOM can decompose by a few different pathways. So in the presence of oxygen, there's this direct aerobic decomposition of DOM that produces carbon dioxide. If oxygen is depleted, we can introduce the iron cycle, where we have an iron reduction reaction, which is supporting the decomposition of DOM also to CO2, which also converts iron 3 to iron 2 ions. And that then allows us to introduce this iron mineral element where we have iron oxides, which are being dissolved under reducing conditions into iron 3, which also then allows us to integrate a feedback on pH. And then there's also a methane production pathway, which starts to ramp up when this more advantageous iron reduction pathway is depleted. And so here are some preliminary simulations from this model framework compared against some incubation measurements from the NG Arctic project that primarily were Zhanshu Zhang's work. So the line here is the model, and I'm going to look first at so if we look at the iron part of this, we can see that the model is reproducing this increase over time of iron 2 concentration in the water, and the model is simulating that as basically due to the depletion of this iron oxide mineral, which is being converted into that more soluble iron 2 as this iron reduction process goes on. Because there's a pH interaction with that, this also causes pH to increase over time, which I think is a nice sort of chemical effect to be able to integrate that most, I would say, soil organic matter based models are not really looking at dynamic pH. And then there's also some changes in CO2 flux, which is higher when iron reduction is going really fast and then decreases over time. And then methane production increases over time as that iron reduction gets depleted. So that's the demonstration of the model. And so a question that I had about this was, because we have this iron interaction, what happens if you change the hydrological pattern of the soil? So here I'm comparing two simulations where they both spend the same amount of time under oxic and anoxic conditions, but the one on the left has one longer oxic period and then a really long anoxic period. And the one on the right is going through more frequent cycles. So there's shorter periods when it's oxic. And so under oxic conditions, we're oxidizing the iron 2 back to iron 3. So if iron 3 is this resource that gets depleted over time as the microbes use it to decompose organic matter, the oxic period basically refreshes that resource. So you have this increase in iron 3 during the oxic period, and then it declines over time through iron reduction. So if we zoom out and look at what's the interaction with between that iron process and the greenhouse gas fluxes, now we can see that this period of iron reduction correlates with lower methane fluxes and a little bit higher CO2 fluxes. And so looking at these two different patterns of inundation and oxidation, in the longer cycle, we basically iron 3 gets depleted and then methane is pretty high for most of that simulation. In the simulation with more repetitions of these cycles, now we're refreshing the iron every time. And so we have more of these periods when methane flux is suppressed by this continuing iron reduction reaction. Because we're specifically simulating the mineral part of that iron cycle, also we can also look at pH and how that affects the solubility of that iron mineral. And so that allows us to look at basically it controls the magnitude of that iron methane interaction. And so when pH is lower, the effect of iron availability on methane flux is much stronger. So this allows us to look at sort of contrasting pH effects on this process, which I think was pretty interesting. And then zooming out again, if we sort of average over these and say, basically, what's the net greenhouse gas impact of a soil being underwater for a long continuous time period versus going through cycles of oxidation and flooding. Going from left to right here, this is just dividing up into how often the soil cycles through oxic and anoxic conditions, and it has a significant but sort of not that strong looking effect on carbon dioxide flux. And I want to point out that the total amount of time that the soil spends under oxic conditions is the same in all of these. So this increase in CO2 flux is not due to more oxygen availability. It's due to just the fact that iron 3 gets replenished under these oxic cycles. And so there's CO2 production that's being supported by iron reduction more when there's more cycling of the hydrology. And so also we get this big decline in cumulative methane production when there's more oxic anoxic cycles. And this interacts with pH, where the yellow dots are the highest pH. And so under low pH conditions, there's a super strong effect of having more hydrological cycling on suppression of methane. So I think these are really promising modeling results for looking at these Arctic soils. And yeah, I'm really interested to take these out of the incubation domain and look at some more realistic like field data when I have the chance. That's kind of the next step for this work. I'm going to step into another project that I'm working on. So this is a project that's supported by a early career award, which started last summer. And the focus of this project is looking at coastal wetlands and also incorporating a lot of these redox reactions. And so what's interesting about these coastal systems is we have to expand from this iron-focused network into a much larger sort of full redox ladder of processes where we have tidal influence that's very dynamic. So we have oxygen fluctuations, we have high nitrogen inputs in a lot of coastal wetland systems from river flows that have high nitrogen loading. We have iron inputs also from river flows. And then the coastal saltwater influence also brings in a lot of sulfate, which supports sulfate reduction as a biogeochemical process. And then methane, again, comes down at the bottom of the redox ladder when all of these other more preferential terminal electron acceptors have been depleted. So this is sort of a preliminary plot just from a simple P-Flowtran setup showing how we can simulate the sequential depletion of these different terminal electron acceptors and then the ramp up of methane production after they've been depleted. So that was kind of a proof of concept. And what we have going on now, which is work that's mostly been being done by Jiazhe Wang, who's a postdoc working with me on this project, is she has a simulation set up with a full tidal cycle where we have, you can see the approximately twice a day high and low tide, and then superimposed on that we have these neap and spring tide cycles. So we have a really dynamic hydrology, which also is a combination of water level and saltwater versus freshwater influence. And so she's starting to simulate this multiple layer column of sediment in a coastal wetland where you have the top layer, which is exposed to the air under some conditions where oxygen is really dynamic depending on where it is in that flooding cycle. And then we have it feeding into nitrate concentration and then also tracking things like chloride and sulfate concentrations. And beginning to see some of these interactions where you have sulfate being depleted lower in the profiles. And in these simulations, it's being supplied by sort of over the top saltwater flooding, and methane production occurring in this profile, but only at the layers where sulfate has been depleted. And I think these simulations are, this whole project is developing in a really exciting way. I'm excited about the direction that's going in terms of being able to look at sort of what happens over tidal cycles and seasonal cycles of saltwater and freshwater influences. And then last, I want to talk about a upland focused application of these type of carbon and other element redox interactions. This is a project that's being led by Beth Herndon with some ORNL money. And the basis for the questions in this project is that particularly in the decomposition of lignin, manganese can play a really important role. So the microorganisms that decompose lignin have one of the enzymes that's really important for lignin decomposition is called manganese peroxidase, and it essentially uses manganese ions, which are really highly oxidizing as these like superoxidant little bonds that blow up lignin compounds and other like really complex biopolymers. And so there's some pretty strong evidence in the literature that the concentration of available manganese for these manganese peroxidases is a strong control on the decomposition rate of lignin. So we can model this by coupling together the manganese and the carbon cycles where lignin decomposition then requires this redox transformation of manganese, which then is in equilibrium with a manganese bearing mineral in the soil. And this feeds into the plant part of the cycle where plants are actually taking up manganese with their roots and depositing it in their leaf litter. And this builds all kinds of interesting connections between bioavailability of manganese in the soil and decomposition of litter. So these are just some preliminary simulation results. We can compare some model simulations of litter mass loss to manganese concentration in the litter. And then these are looking at basically the interaction of soil pH, which controls the solubility of manganese-bearing minerals, and essentially the average oxygen availability, or like the average redox state of the soil, where if we have more time spent under anoxic conditions, there's a higher manganese availability, because reductive dissolution of minerals is a main source of manganese that's bioavailable to plants. And so we can map out how under lower soil pH conditions and more often anoxic conditions, leaf litter manganese concentration is higher, and then this drives the organic matter stock in the litter layer to be lower in these model simulations, because manganese availability is promoting lignin decomposition. So to wrap all this up, these are all sort of like offline simulations right now in pFlowtran, but another big part of this early career project that I'm working on is upgrading this interface to couple all of these pFlowtran simulations into the ELM land surface model. And so this interface is, I would say 75% done. And so I'm pretty excited to be able to basically take these pFlowtran simulations, couple them into ELM, and then look at how some of these carbon and other mineral interactions then propagate into the carbon cycle. I think we're close to the end of our hour, so I'm gonna finish up here. But if anybody has any questions or would like to discuss sort of what's interesting or what's difficult or what's promising about trying to couple together the carbon cycle with some of the other elemental cycles and models, I'd be happy to talk more about it.